also known as daltonism, is a sex-linked recessive disorder that is carried in the X chromosome. In which diagnosed individuals cannot distinguish the difference between certain pairs of colours. The study was conducted by Catherine Mieszko et al. In aim of regaining colour vision in dichromatic squirrel monkeys. Most female Siamese squirrels are born with colour blindness, while all male squirrel monkeys do not have the three photopigments that are essential for trichromatic vision and are therefore colourblind. In primates, it is normal to have three cones, which are small, medium, and long cones. However, in primates that have colour blindness, they do not possess medium or long cones. M and L cones are wavelength-sensitive photopigments. An experiment was conducted in the USA in aim of regaining colour vision in squirrel monkeys. Firstly, a colour vision test was conducted prior to treatment, and dichromates who were missing the M or L cones failed to distinguish colours in the blue/green and red/violet section from grey. The monkeys had to distinguish different coloured dots that had different sizes and brightness levels. They were surrounded by similar grey dots. When colours were touched, a positive sound would be exerted along with a sweet drink given. However, if a grey colour was touched, a negative sound would be played and no treat or fruit drink would be given. Two dichromatic monkeys performed three colour tests which consisted of 16 hues over a period of 4 to 6 months, which can be seen in figures B and C. The results show failure in recognising blue and green and red and violet hues. In order to repair and insert the missing allopurin gene, scientists inserted a human allopurin gene into a virus which transported the gene what is needed to allow the squirrels to have trichromatic vision. Allopurin is a light-sensitive cone pigment found in the retina, converting light energy into electrical chemical signals interpreted by the brain. A human allopurin gene was delivered to the photoreceptor layer by subretinal injections. Several of these injections were performed. The two dichromatic squirrel monkeys were then injected 3 times with an allopurin-encoded virus which produced a homogeneous region. The results for this procedure can be seen in figures 2B and 2C. Experimental results were tested to see whether the gene therapy worked. This was performed by observing the animals' behaviour when different colour pictures appeared. Before the injections were given, they could not distinguish the difference between certain colours, whereas after the injections were inserted, after a period of 20, 40, and 72 weeks, seen in figures C and F, the squirrel monkeys were now trichromatic, and they could be able to distinguish red from green and so forth. This can be seen in figures 3A, where wavelengths of 490 nanometers are represented by the circles, and wavelengths of 554 nanometers are represented by the squares. On the left, we can see an example of a dichromatic primate, and on the right we can see an example of a trichromatic female squirrel monkey. The updated article had similar findings and methods. Figures 3B and 3A show that after the treatment, the squirrel monkeys now lacked a spectral neutral point and have become trichromatic. Overall, as seen in the improved results, colour vision can be restored through the use of gene therapy just by adding missing cones. Thank you.